Welcome to this webinar. This webinar is brought to you by BioServe, which is a REPROCELL company in Hyderabad, India. My name is Dr. Kishor Bhatia, and I serve as a consultant for REPROCELL, along with other areas where I have responsibilities, such as the Chief Science Officer for Lantern Pharma. I also continue to do research as an Adjunct Investigator with the NCI. This slide gives you a general glimpse of the REPROCELL group of companies, which are present in USA, UK, India, Japan, where the headquarters are in Japan. Our various areas include biorepository, drug discovery, stem cells, CRISPR and Cas9, development of cell lines, as well as next generation sequencing. Our focus of discussion today is precision oncology. The suite of assays that REPROCELL has developed for this area are termed as PreciONC. Those of us who are actively involved in conducting cancer research and are involved in the area of oncology have witnessed during the last decade, as evidenced by presentations, talks, papers, and practice, that there's a clear evidence that both research and clinical oncology has entered a new era. During the past decade, many strides have been made in moving towards more precise diagnosis and treatment. This direction was in large part catalyzed by a better understanding of the genomic landscape of tumors, which in turn was made possible by lowered costs and enhanced platforms for sequencing of DNA. Rapid discovery of multiple non-overlapping driver mutations, the development of tyrosine kinase inhibitors that proved clinically effective in cancer cells, such as lung and melanoma, led to clinical assays, first based upon PCR platforms, which allowed rapid and inexpensive identification of biomarkers. But such PCR-based assays raised expectations, which in several areas have been met, and these induced the advent of more comprehensive molecular profiling. While our ability to match genomic aberrations to specific treatment strategies and choose the right drugs from an ever-increasing drug armamentarium continue to improve, the reality is that such approaches still remain unavailable to all patients globally. A clear unmet need exists in many geographical areas, and this unmet need is not just a consequence of economics and costs. As you will likely realize, the delivery of precision oncology is quite dependent upon some key foundations, such as the ability to genomically characterize tumors, the ability to interpret what the genomic analysis indicates, and the availability of drugs matched to such characterized abnormalities. In India, REPROCELL seeks to provide a solution by providing accessibility to platforms that allow a better characterization of the genomic features of the tumor, so that patients and oncologists can best utilize the available drug choices. So, if one had to define precision oncology, at the most fundamental level, it can be defined as the application of molecular profiling to allow treatment tailored to the genomic characteristics of an individual's tumor, and the ability, therefore, to select drugs that are most efficient and least toxic. And this new era, therefore, moves away from the previous paradigm of treating tumors with the same blanket chemotherapies. The availability of knowledge of the features of the tumors allows approaches to use drugs that will target the tumors selectively. Again, the critical requirement, however, is that tumors be assessed for the presence of mutations, for the presence of genetic aberrations, and or transcriptome analysis that provides both the knowledge and the justification for choosing the right drugs. The drugs are matched to what is often known as a molecular target. A molecule that is intrinsically associated with a particular disease process, such as etiology, progression, and or drug resistance, is the molecular target. It also needs to have evidence that intervention of this target will cause an alteration of the disease process, and that this alteration will result in positive and therapeutic outcome. So the entire intent of genomically characterizing the tumor is to identify the molecular targets for which there are drugs available. And therefore, these alterations become actionable because now one can take a specific drug to intervene in the etiology of the disease or the progression of the disease. Most of the actionable alterations come from the presence of somatic mutations. And as you all are aware, the concept of somatic mutations is as the foundation for cancer development, therefore underpins the entire approach of precision oncology. Actionable drug targets are not new. Early on, such as in 1998, the understanding of the presence of a BCR-ABL translocation in CML led to the development of a drug that specifically targeted BCR-ABL or the tyrosine kinase of that moiety, and this drug was imatinib. And matching of imatinib for the treatment of CML came about by using PCR assays or RT-PCR assays to show the presence of the BCR-ABL translocation in CML. Therefore, this genomic test had clinical utility. But in general, genomic tests with clinical utility should be predictive of treatment response from a targeted agent. Another example in solid cancers comes from the test of HER2 positivity, either earlier with FISH or with immunohistochemistry, to demonstrate the overexpression. And this overexpression of HER2 matched with response of tumors to drugs such as trastuzumab. Lung cancer has now become the paradigm of precision oncology. Actionable biomarkers of non-small cell lung cancer have made this disease an excellent example and model of precision oncology. The application of precision oncology in this disease has dramatically impacted survival in lung cancer. And these genomic tests remain relevant at diagnosis, during therapy, as well as during the time of progression. In this slide, what is clear is how this evolution has occurred. Early in 1987, there were not that many alterations of lung cancers that were identified or known. The most prominent alteration in lung cancer that was known was mutations in KRAS, but that defined only a small percentage of lung cancers. The vast majority of lung cancers were not matched to specific alterations. This began to change in 2004 when mutations in EGFR were discovered in a proportion of lung cancers, which varies now from 20% to more, depending upon geographical location and so on and so forth. The presence of EGFR mutations allowed the development of a drug that is targeted to such EGFR pathways, making this mutation an actionable target. Now, there are many more genomic alterations that have been identified in lung cancer, including EGFR sensitizing mutations, translocations involving ALK, ROS, and mutations in many other genes. An excellent example is NTRK, and I will discuss a little bit more about NTRK in some of my later slides. So now the paradigm of treating lung cancers has changed. Previously, lung cancers, irrespective of what mutations they had that were known or not known, they were treated with a blanket chemotherapy of platinum doublets. The new paradigm is to first define what actionable targets are present in lung cancer and treat these lung cancers with that targeted therapy, resulting in large and clinical meaningful benefit. The reason that such benefits are derived is because for each of the actionable target, whether it be mutations in EGFR, alterations of ALK, alterations of ROS, so on and so forth, there are matched drugs that can inhibit that pathway. I think it's important also to understand why genomic alteration monitoring is relevant even during treatment. Some of the drugs, such as erlotinib, which is targeted to EGFR mutations, eventually fail to respond because such actionable targets get modified and there is development of resistance to certain targeted therapies. An example of this would be evolution in lung cancer exposed to erlotinib, where EGFR mutations at T790 at the residue 790 occur. These mutations are what are known as gatekeeper mutations, but identification of these alterations then allow a choice of another tyrosine kinase inhibitor, such as osimertinib. Our identification of actionable targets in lung cancer continue to evolve, not only in the identification of genomic alterations, but also in parallel, the development of drugs for alterations that have been known, but until now have remained undruggable. A good example of this is mutations in RAS. We have known for quite some time that KRAS is mutated in 20 to 30% of many cancers, including colon, lung, and pancreatic cancers. While RAS had remained untargetable, so there was no drug that targeted KRAS mutations. Nonetheless, testing of RAS mutations remained important and relevant. An example comes from colon cancers, where colon cancers that had EGFR based alterations and were therefore treated with EGFR based therapies, the presence of RAS mutations in such colon cancers indicated that EGFR based therapies might be less effective in these colon cancers, and therefore testing of RAS mutations was relevant even at a time when there were no specific drugs available for RAS. However, recently, a drug that targets a very specific KRAS mutation, that is at codon 12, the G12C mutation, has been approved by FDA. So now we have another 13% of non-small cell lung cancer adenocarcinomas, where another actionable mutation can be defined. So it's the specific identification of the alterations in the tumor that allow personalizing therapy. And just to summarize the status in lung cancers, where there are specific EGFR alterations, often activating deletions in exon 19 and point hotspot activating mutations in 21. I already discussed with you the possibility that during the evolution, during treatment, one might acquire resistant mutations in exon 20, and therefore change in therapy could be indicated. Rare mutations also occur in EGFR, and these rare mutations, such as those with exon 20, may not respond to other TKI inhibitors, and there are specific TKI inhibitors such as poziotinib that are used for patients that have exon 20 insertions of EGFR. For patients that have alterations in MET, which often is a MET exon 14 skipping mutation, there are choices of tyrosine kinase inhibitors that can be used. Lung tumors also have fusions that involve the NTRK gene, either NTRK 1, 2, or 3, and there are tropomyosin receptor kinase inhibitors that have been approved for those tumors that have such alterations. And as I discussed with you, now we also have drugs for the specific mutations in KRAS, making the identification of KRAS an actionable drug target. I mentioned to you a while ago that I will discuss briefly the case of NTRK fusions. So these are genes called as the tropomyosin receptor kinase family of genes, TRK, TRKB, and TRKC, which are encoded by the neurotropic receptor tyrosine kinase. Now, TRK fusions lead to the expression of a chimeric oncoprotein And this chimeric and oncoprotein independently activates the kinase pathway driving oncogenesis. A specific aspect of TRAC fusions are that these actionable drug targets are present not just in lung cancers, but are present in many adult and pediatric cancers. So although these alterations are rare in any one given cancer, together several cancers make the this drug target a key feature of personalized oncology. Here is a graph from a Lancet Oncology paper by Hong et al. And on the y-axis, you see a change in tumor size. This is a waterfall plot, and the key aspect of the key message in this slide is that the presence of NTRK fusion in various histologies, including appendix, cancer of the unknown primary, lung, salivary gland, melanoma, pancreas, thyroid, so on and so forth, allows a drug like larotrectinib to positively affect responses irrespective of the histology of the tumor, but totally related to the presence of a TRAC related genomic alteration. You can therefore understand that the TRAC inhibitors, and particularly larotrectinib, was approved as an oncology drug that targets a key genetic driver of cancer rather than a specific type of a tumor. So similar parallel genomic alterations have been identified in cancers beyond lung cancer, allowing one to develop personalized oncology approaches. Obviously, again, I think it's important to state that in order for these approaches to be available to patients, the identification of these alterations is a key aspect. In breast cancers, amplification of ERB-B2 allows the choice of anti-HER2 therapy. But there's also a fraction of breast cancers that may not have amplifications but have mutations, and this allows for a different choice of TKI inhibitors. We are all familiar with the increased risk of familial breast cancers because of mutations in BRCA1 and 2. And the presence of mutations in BRCA1 and 2 allows one to make choices between several available PARP inhibitors. A proportion of breast cancers also have mutations in the PI kinase pathway, and particularly mutations in PIK3CA in exons 7, 9, and 20 allow for a very specifically matched drug to this genomic alteration. If there are mutations in the estrogen receptor 1, which is rare in primary but common in metastatic cancers, that indicates resistance to aromatase inhibitors and therefore allows specific choices of therapy based upon this feature of breast cancers. I already discussed with you about NTRK gene fusions. Again, these are also present in a proportion of breast cancers. Some breast cancers show evidence of microsatellite instability. And microsatellite instability has been matched to a better response to immunotherapy. Breast cancers that have mutations in PTEN and ACT1 are likely to benefit from drugs that are inhibitors of the ACT pathway. Moving from breast cancers to metastatic colorectal cancers, the use of cetuximab and other EGFR-directed therapies have been approved for EGFR-expressing metastatic colorectal cancers. As we discussed earlier on, the identification of mutations in RAS is critical, particularly in EGFR-expressing colorectal cancers, because the simultaneous presence of mutations in RAS, along with an activating EGFR, predict that EGFR monoclonal antibodies are unlikely to effectively work in those cancers. And therefore, assessment of mutations in RAS is very useful in precision oncology approaches in colorectal cancer. Microsatellite instability often is present in several colorectal cancers. It comes from a deficient mismatch repair system. And the most commonly affected mismatch proteins include MLH1, MSH2, MSH6, and PMS2. And some of these aberrations also increase the risk, familial risk for colorectal cancer, as we'll see sometime later in this webinar. Microsatellite instability, high status is present in 15% of colorectal cancers and can be identified using PCR-based test. As I mentioned to you earlier, the presence of microsatellite instability status corresponds well with a tumor responsive to immuno-oncology or immunotherapy approaches. And the reason for this is because tumors that have microsatellite instability likely generate new antigens which can enhance the effect of immunotherapy agents. And therefore, testing for microsatellite instability has become valuable because it predicts the efficacy of immunotherapy agents such as nivolumab. Certain proportion of colorectal cancers carry mutations in genes such as BRAF, particularly the V600E mutation, suggesting the utility of RAF inhibitors in treatment. And TRAC fusions are also present in a small percentage of colorectal cancers, and in those cancers, TRAC inhibitors would be indicated as a choice of drug. Prostate cancers are often defined alterations in DNA repair genes. Several of these DNA repair genes, including PALK2, RAD51, D, RAD51C, ATR, MRE11A, are altered in prostate cancers. But some of the more well-known DNA repair genes, including BRCA1, BRCA2, and ATM, that would make the tumor deficient in homologous repair mechanisms, are also present in prostate cancers. And such prostate cancers can therefore be matched to use of PARP-I inhibitors, particularly prostate cancers where the alterations in BRCA1, BRCA2, and ATM are are present as germline mutations. PTEN alterations would suggest the combination of ACT inhibitors along with androgen-based therapies. In metastatic gastric cancers, one finds the presence of ERB-B2 amplifications. One also finds the presence of MSH microsatellite instability status, and therefore these two features again allow one to match the alterations with specific drugs that are targeted to these features. Other alterations include NTRK fusions, but there's also a proportion of gastric cancers where you have alterations in DNA repair genes, invoking the use of synthetic lethal agents that are particularly targeted at alterations in BRCA1, BRCA2, and ATM. Hotspot mutations such as those that we talked about in breast cancer, the PI kinase hotspot mutations, have also been reported in gastric cancers. This contribution of DNA repair mutations extend to several cancers. We discussed their presence in breast cancer, in colorectal cancer, and in prostate cancer. But these have also been shown to be present in pancreatic cancer. And particularly if alterations in BRCA1 and 2 are present as germline alterations, there is data available that treatment with synthetic lethal agents such as the PARP inhibitors, olaparib, result in a longer progression-free survival. Whether olaparib provides benefit if BRCA1 and 2 are present as somatic mutations in advanced pancreatic cancers is still unclear, but there are some prospective trials which do suggest an increased response. Pancreatic cancers that have NTRK fusions would obviously benefit from the respective targeted therapy. So we spoke of several cancers that have mutations that affect DNA repair pathways. And one particular pathway is featured quite prominently in many cancers, and this is the homologous recombination pathway. And so these cancers that are deficient in homologous recombination, which include breast cancer, ovarian cancer, pancreatic cancer, and prostate cancer, appear to respond well if there is a known alteration in BRCA1, BRCA2. Other drugs in the PARP inhibitor family include niraparib, which has evidence in ovarian cancer, rucaparib, which also has evidence in ovarian cancer, and talazoparib, which has evidence in breast cancer. We spoke about the microsatellite instability pathway. And this microsatellite instability comes from alterations in the mismatch repair pathway. And as we discussed earlier, microsatellite instability appears to correlate with a better response to immunotherapy agents. And this is a graph from a paper by Yoshua et al. And what this graph indicates is that from various histologies, if you look at the objective response rate of the tumor, it appears to correlate quite well with the median number of coding somatic mutations per megabase. And in general, one can approach this feature of the median number of coding somatic mutations per megabase of the tumor by using an assay that measures what is known as a tumor mutational burden. So the higher the tumor mutational burden, the better the response rate to immune checkpoint inhibitors. And therefore, assaying for tumor mutational burden is another important consideration one could have if one has to choose the use of immune checkpoint inhibitors. But beyond MMR deficiency, there are also considerations that can be given to looking for mutations in those genes, such as the DNA polymerase genes, POLE, POLD1, that affect proofreading activities of DNA synthesis, allowing for allowing for the tumor to incur additional mutations. For example, the mutation in POLE at amino acid 286, the P286R is now a recognized pathogenic variant that correlates with better response to immunotherapy. So in general, I think what appears to be documented is that if one looks for actionable genomic alterations, as many as 70 to 80% of patients will have alterations that can be matched to drugs in the available drug armamentarium, and therefore identification of specific features of the tumor, specific genomic features of the tumor, is likely an important aspect of treatment approaches as we move forward. I'm going to spend a little bit of time on hereditary cancers. Precision oncology allows one the ability to identify germline and familial genetic alterations that might increase cancer risk. Use of targeted screening and early detection allows one, therefore, to prevent development of advanced cancers. And since now there are many different possible genes that affect at various penetrances the risk to develop cancer, comprehensive panels have been developed that include such genes. An example would be BRCA1 and BRCA2, but as we will discuss, there are genes that go beyond BRCA1 and BRCA2. Furthermore, as we talked about pancreatic cancer, the presence of germline mutations in some of these genes allows one to define whether a targeted therapy, such as for pancreatic cancer, if BRCA1 and BRCA2 are present in the germline, the use of PARP inhibitors allow a better response can be defined. So if you look at familial syndromes, particularly for breast cancers, there are several familial syndromes. The more well-known is the hereditary breast and ovarian cancer syndrome, which results from germline mutations in BRCA1 and BRCA2. Again, to remind you, BRCA1 and BRCA2 are DNA repair genes in the DNA homologous recombination pathway. The Lee-Fromeni syndrome, which results from alterations in P53, Cowden syndrome, which results from alterations in PTEN, the hereditary gastric cancer syndrome, which results from alterations in cadherin genes, the Peutz-Jeghers syndrome, which results from alterations in STK11, and the Lynch syndrome, which many of you are familiar with in the context of colorectal cancer, which results from alterations in the mismatch repair DNA pathway genes. Speaking of colorectal cancer, the syndromes that increase the risk for colorectal cancer would be the MMR, the mismatch repair pathway genes, MLH1, MSH2, so on and so forth, which occur in the Lynch syndrome. Then amongst the two polyposis groups are the familial adenomatous polyposis syndrome resulting from alterations in APC, the MYH associated polyposis syndrome resulting from alterations in MYH. Peutz-Jeghers syndrome also increases risk for colorectal cancer syndromes, and as we discussed, this syndrome results from alterations in STK11. Juvenile polyposis syndrome results from alterations in SMAD4. SMAD4 is a chromatin based pathway gene, and the Cowden syndrome, which we discussed earlier, resulting from alterations in PTEN. Pancreatic cancers have also shown to have familial risk, and syndromes where there is alterations in BRCA1 and BRCA2, such as the hereditary breast and ovarian cancer syndrome, would have an increased risk, lifetime risk of pancreatic cancer. Other syndromes where you find an increased risk of pancreatic cancer include Lynch syndrome, Peutz-Jeghers syndrome. But clearly, the risk for familial cancers, including breast and gynecological cancers, is associated with alterations not just in BRCA1 and BRCA2, but because the pathway that BRCA1 and BRCA2 are involved with include many other genes, including ATM, RAD51C, RAD51D. It is quite clear that alterations in these genes would also increase risk to hereditary breast and or other gynecological cancers, and therefore comprehensive panels that include many of these genes have been developed. Some of these panels are limited, such as the one shown above, which is 18 genes, but many experts have now expanded the number of genes associated with hereditary breast and gynecological cancers, and additional genes such as SMARC, A4, STK11, and NF1 are included in these panels. So what I think our previous discussions, one of the key things it points out is that personalized oncology derives benefits from the use of next generation sequencing, because there are multiple alterations that can be present in a tumor, and some of these alterations would allow for matching of the alterations to specific drugs or defining what is the actionable alteration. So NGS provides the ability to perform a comprehensive test, one sample, one assay, one report, and can detect all different types of genomic alterations, be they mutations, fusions, or copy number variations. In addition, unlike traditional methods, next generation sequencing has higher sensitivity. It's important to look at certain considerations in understanding what is the right NGS test for your patients. Should one go for large comprehensive gene panel versus targeted actionable gene panel? Some of these considerations might include what are the sample requirements for these two types, what is the turnaround type, and what is the cost. Clearly, cost for smaller panels might be lesser, but larger panels are likely to give you more clinically relevant information. But some of the things to consider are that as you use comprehensive panels, you're likely to identify variants of unknown significance, and such incidental findings can be challenging. The turnaround time for smaller panels might be shorter compared to the larger panels. BioServe REPROCELL provides several different types of panels, including the gene fusion lung cancer panel, which include the ability to detect rearrangements of ALK, RET, ROS, and the NTRK genes. REPROCELL also provides integrated tumor panels that allow you to define the tumor mutation burden and provide a comprehensive assessment of over 409 genes. One could include tumor mutation burden along with MSR testing to get a better sense of the use of immuno-oncology agents. So in summary, there's a plethora of oncogenic driver mutations currently known, with a growing number of targeted therapies to match. Next generation sequencing obviously simplifies the testing algorithms, and several large solid tumor panels are available that can yield actionable results. If there are any questions, if you have any specific needs related to precision oncology, here is some additional information on REPROCELL. I hope you enjoyed this seminar. Thank you so much for your time.